So thank you very much for the kind introduction. It's a very interesting experience to be here. First, an admission, I'm not a plant breeder and I don't know very much about plant breeding. So treat me lightly, please, with the questions. But I am interested in G by E and exploiting G by E to understand the implications of climate change for our agricultural systems. The question is how to do that, and what will our arable systems look like in the future? If you have a breeding company, like what are you going to need to breed for in 20 years' time? It's not a silly question. If you're a policymaker, what is the landscape going to look like? Who are the winners and the losers going to be? Because there's always winners and losers, right? So how to understand how climate change might affect a crop? I'll just give you a case study in winter rapeseed, European winter rapeseed. So most of our understanding of effects of climate change on crops come from studies with crop models, like process-based crop models. And they're supposed to capture what plants do in the real world, and people take a climate model, crop model, marry up the two, get output, write paper. I just put one example of this. They say, oh, actually, it didn't work very well. All the crop models gave us different results. And really, they conclude they need more crop modeling. Now, I'm going to try to essentially show you a different approach to tackling this question. And so, what are the issues here? There's a lack of model agreement, really, in the outcomes climate-based simulations for a lot of crops. That's the first problem, but it's not the most important problem. And there's actually little or no effort spent on testing the predictions of crop models. So it's very interesting. I consider crop model as a hypothesis-generating algorithm rather than as something that is really going to tell you something that you know with any certainty. So keep that in mind. The third point I put on this slide is that there may be biology that we don't understand that's relevant. And I'll show you an example of this. And of course, if we don't understand it, it won't even be represented in a crop model. Just think about that for a moment. So crop models are a bit hubristic in that sense. They assume that we know perhaps a lot more than we do about crops and their biology. And the fourth point, I think, is also really important, is the pace of climate change is such that there will be breeding, and with breeding can come adaptation. So a crop that is grown in a location today might not behave like a crop grown there in 40 years' time, because in between time, the breeders will have done some breeding. Ooh, there we go. Some of you maybe. Right, hopefully that's not important. Right, so I'm going to explain to you something probably different to the way you hit it before. Actually, climate change is quite advantageous as a system perturbation to work on. It has two key advantages, right? First is it's highly predictable. And the second is it occurs over decadal timescales. This is brilliant. Compare this to the invasion of Ukraine by Russia. This doubled oilseed prices within weeks. Right? And to do that by breeding, you would have needed hundreds of years of effort, to generate the same change in farm incomes. In fact, you probably never succeed. So some perturbations are very quick, some perturbations are very slow. Take banning of neonicotinoid pesticides, so seed treatments in Europe. A stroke of a pen, and it cost the UK rapeseed industry £510 million. That's about the same as you might make from 175 years of breeding as a farmer. So climate change is really nice for that. So the breeding gains take place over similar timescales, like one could argue, right? But that's, of course, assuming that breeders pull their fingers out and do some breeding that makes a difference on farms. And if you look at the green line on this chart, just shows UK rapeseed yield over time. Actually, statistically, we can only detect one yield gain event over the whole period. So whether that's one gene, we don't yet know. But all that G by E you see, and all those genetic gains in breeding cycles don't necessarily translate to measurable genetic gains when you scale up to commercial seed productions and give seeds out to farms. That's always worth bearing in mind, that there's an assumption that those genetic gains you see in field trial, that they scale. Sometimes maybe they do not. Right, so it's important that we realise these breeding gains in order to have a chance of mitigating climate change, but it also has to be, the adaptation has to be attainable genetically. And so this is what this talk is about. To understand a bit more about it, you have to know a little bit about winter rapeseed, and I'm aware some of you probably don't know very much about European winter rapeseed. It's normally sown in August or September, so late summer or early autumn. And really, you consider sort of range of crop types from, in the west of Europe, what we call these maritime types that have the main problem is a risk of low winter chill, and that's really going to be an important subject to the talk. Whereas in the east, you have a separate problem, is that you can have very cold weather, and if there's no snow cover, this can actually lead to frost damage and something which rapeseed breeders call winter kill. So you're balancing these two crop types within winter oilseed rape by breeding for different, and many companies will have a different breeding program, for instance, for west and east or something like that. So back to climate change. The other thing to realize is that although you hear a lot about mean climate change, actually it's not uniform over the year where we see gains, for instance, in temperature. So here I'm looking at temperature, just using the Central England temperature series, the world's longest running temperature series, just looking month by month, how much change has there been. You can see that the change is concentrated in autumn and early winter. And of course, the reason for this is that the sea aggregates changes in temperature throughout the growing season and stores that energy and releases it when it gets cold. So it's really important to note that you're not getting a uniform change over the year. And of course, temperatures during the winter have already increased a reasonable amount. This is just showing winter temperatures. So how to get at this problem? So we really want to know, can we breed for resilience to weather variation? What weather should we breed for resilience to? And how should we go about it? And to do this, we turn primarily to analysing national list trial data. And this took some time, but we got somewhere in the end. And the key thing you need to know about winter oilseed rape is that it has this chilling requirement which leads to flowering. So it's sown in the autumn, chills over winter, leads to flowering. And the one gene you need to know about is this gene FLC, which was discovered in Arabidopsis, which confers the chilling requirement. And it starts off high expressed and it's silenced by chilling, and then the plant flowers. And that's really the Arabidopsis model. That's the only genetics that you will need, and there's only going to be one cross in this whole presentation. But on the left here, you can see the relationships that we uncovered, which we think are interesting. We saw this really strong positive correlation between October mean temperature and yield. You can see it's quite a nice scatter plot. And then a negative correlation between December temperature and yield. And of course, one thing we were interested in is whether this is something to do with vernalization. So we also did some experiments like transcriptome time series in the field, and this is just looking at genes that are involved in flowering. And really what we found is this, that these sort of FLC genes that are silenced by chilling, the red is the high expression and the blue is the low. And you can see this series starts in September, and very quickly these FLC genes, they disappear. Their expression goes down, and genes to do with flowers come up. And actually, when we started dissecting these apexes, you can see here, so this is what an oilseed rape shoot apex looks like when it comes into flower. Now, these plants look like they're growing vegetatively and producing leaves, but actually, when you dissect it down, at the beginning of November, they're already producing these little flowers, and this is what these balls are here. These are the flowers, and you can see them growing. So already in November, they're producing flowers. So what it turns out then is that before they start producing flowers, we have this positive correlation with temperature and yield, and after they start producing flowers, we have this negative correlation. And really, this inflection point occurs in these relations. It occurs exactly when they start making flowers. And that's the fundamental biology which we understood. The one issue with this is, of course, that if this chilling is somehow important because it affects the crop, and I'll try and persuade you that it does, then why do they need chilling after they start making flowers? Because really what's supposed to happen is that they chill, they vernalize, and then when vernalization is complete, they start making flowers, right? So this is kind of upending our understanding of what chilling is doing in an arable crop. And you can see this quite clearly when we look at these FLC. So Brassica napus has several FLC genes. And like I said before, red is the high expression and blue the low. And the black line is when they start making flowers. And so you can see that a lot of these FLC genes, they chill apparently before they start making flowers. But then there's these two at the bottom that hang around afterwards, right? And this is basically the key to the talk. So what is this going on? Like, how do we investigate what the plant is doing in the field at this time of year and the effects of temperature? So what we did was we developed a system for actually warming the plants in the field. This is a non-stressful warming. It's not cooking plants or anything like that. So we're delivering a gentle heat that's going to warm the crops by about 5 degrees Celsius. And we do it with these cables, which are designed for heating gravel beds in horticultural systems. If you fly a drone with a thermal imaging camera over a trial, it looks a bit like that. So you can see the warming, and the warming turns out an average of about 5 degrees. But it does vary a lot. If it's very windy, the heat blows away, right? So this is a real world scenario. This is not a perfect system. So what happens if we warm in October? Question one. So the control series of dissections is on the top and the warmed on the bottom here. And you can see the timings and the control ones. This is a different growing season, but they still start making flowers at the beginning of November. And then the warm October, you can see they don't start making these little ball-shaped flower primordia until December. So warming in October by about 5 degrees has delayed floral initiation by about a month, right? That's just quite an interesting phenomenon. So this is what warming in October is doing. And this is associated with increased expression of these FLC genes. Like this one, A2, is this one at the top here, that's silenced before the reproductive transition. So that's what's going on in October. So this leaves a question, right? If vernalization is happening in October and then the plants flower, why do they need chilling in December? And this is fundamentally something that made no sense in terms of our understanding of crop developmental biology. So we did some lab experiments and some field experiments. If you, or at least after they start flowering, you also delay flowering. So even though they're making flowers and they've vernalized, you delay flowering. And you can see this also in the field. So these have been warmed. And you can see the control plot on the left and the warmed one. And the warmed ones, they have a lower yield. And this lower yield is associated with the increased production of a hormone called abscisic acid, ABA. And what this ABA is doing, we have learned, is imposing what we call bud dormancy on the flower buds. So in the same way you get winter dormancy cycles in trees, you're getting the same response in these flower buds in brassica crops. And in fact, we can detect this in cereals, we can detect it in a lot of crops. And this is associated with the failure of silencing of these FLC genes that hang around into floral development. So what we have here is a response that crops have to chilling that no one appreciated existed before. And of course, is really important for crop behavior, but moreover, if it's perturbed, affects the yield. So we've learned that warm autumns, they extend the vegetative phase, and they ensure that floral development, therefore, takes place at a colder time of year. And that's one reason that warm autumns are good. And then floral development at lower temperatures associated with lower ABA levels, earlier flowering and higher yields. And this involves the different types of FLCs. Right, so the question is, can we breed for resilience to this? This is really the topic I want to talk about today. And 16 minutes. Right, okay. So I've got some time. So this we went to what I call, a stereotypic, very basic, because I'm not a breeder, G by E. And we went back to the national list trial data, and because we have a specific window where we know the chilling is important, it's between floral initiation and the winter equinox that we know this chilling is affecting yield. It doesn't matter what the chilling is later, it's particularly in this period. So we can take the temperature variation during this period specifically and put this into a G by E study. And that's the thing with G by E, is you have to know exactly what variable you need to put in. And so we knew exactly what variable we could do the G by E. And so here's a classic, so it's an old plot, like it's created with GenStat, so it's not very pretty, but there's a variety temperature effect and a variety effect. And we picked out this one variety, Castile, which was bred by Monsanto, now part of Bayer. This seems to be the most resilient to variation in the chilling in this period. It's not the highest yielding. Remember, I showed you there's one yield gain event. This was a variety that was cultivated before that yield gain event. So the question is this variety useful for breeding for resilience? This is what we wanted to do. So the next series of slides I show you are going to be comparing two varieties. One we're going to call our standard variety, Campus. This is an elite variety bred by KWS. It's a very nice control variety. We use it in a lot of our experiments because it's a good experimental variety, and it's also, even though it's about 10 years old, its yield and performance is not statistically different from current elite varieties in our hands. So we use a Campus, and then we have Castile. So the first thing we wanted to do is we bring this variety in a lab. All we've done is to try to detect this effect in a G by E study in national list variety trials. Does it really have a different chilling response? That's the first question. And remarkably, it does. So what we've done here is we've chilled Campus or Castile for either 3 weeks, 5 weeks, or 7 weeks, and they're the different colors. And we've chilled them at either 5 degrees Celsius or 15 degrees Celsius. And you can see that if you change the chilling temperature, it makes a big, big difference for Campus in the flowering time. So BBCH51 is buds visible for those of you not familiar with the scale. And actually the Campus doesn't really flower. We get bored waiting for it if you chill it at 15 degrees. But the Castile, really different. The Castile, you bring it into the lab, chills quite happily at 15 degrees Celsius, which is remarkable. So I didn't really believe it when I saw this, that you could just go through a variety trial data and pick out a variety with chilling, resilient to variation in chill, and bring it into the lab and find that it does. And the reason we did these two temperatures is because we wanted to sort of separate autumn and winter chilling, which should take place at different temperatures, right? Because autumn is warmer than chilling. So 15 is kind of like an autumny chilling, and 5 are wintry chilling. So Castile's, this autumny chilling temperature seems sufficient, like nearly, to chill it. So we also did some things like transcriptomic studies, and you can see we get this big transcriptome response to warm winters in campus. All these orange, reds are the warm winter-induced genes. These are all the genes that are involved in generating the bud dormancy and the response to the developmental stasis that occurs. You can see in Castile, this response is really low. Like all these genes, lots of them are still blue. So, and lots of these are to do with ABA. And a lot of G by E, basically in reproductive development, is due to ABA. Is really a take-home message. So, what is the genetic basis of this? And I promised you we would do one cross. So, no surprise, our one cross is Campus Castile. We assembled genome sequence for them, like pretty high-quality reference sequence. And what I'm showing you here on the left is just the coverage, 2 chromosomes. Now, if you're not familiar with Brassica napus, it's an allotetraploid, so it has the genome from Brassica oleracea and also from Brassica rapa. So it contains both these genomes, but they're both quite closely related species, and this is important for what comes next. So we have the A2 chromosome. This is the chromosome 2 that originates from Brassica rapa. And we have the C2 chromosome. This is the chromosome that originates from Brassica oleracea, so your vegetable brassica. And you can see the coverage plot. And the key thing to note is here in Castile. We have this block at the front end of A2 where the coverage appears to double. And at the front end of C2, we have this block where the coverage appears to tend to 0. And why is this? And this is a phenomenon that's known in brassica genetics, and it's called a homologous exchange. And it occurs when you get aberrant pairing. So instead of C2 pairing with C2 during meiosis, you get C2 pairing with A2 because they're quite closely related, even though they're from different species. And then what happens when you get recombination is that you get a segment from A2 transferred onto C2. Now the interesting thing about this, this one switch is where the FLC gene lies. So the FLC A2 from Brassica rapa is one of these autumn chilling FLCs, but the C2, in fact, is the highest expressed winter chilling FLC. So what Castile does is it takes this sort of, so Campus will have your autumn chilling and your winter chilling FLC, and Castile just has 2 copies of the autumn chilling FLC. And fundamentally, it's not the only genetic difference, obviously, between these 2 varieties, and there are other genetic differences which are important in the different behavior, but just for the simplicity, because this is the most important one, I'll focus on this. So here we go, and there are also other genes, of course, in this chromosome segment, and we're still ruling out that some of these are important, but we think this FLC is the most important. And so to our one cross, so we have a doubled haploid population between these 2 varieties, and anyone can have this if they want. And then we score flowering either in the field or in the glass house, doesn't really matter, but it's nice to do it in a few environments just to see how robust these phenotypes is. And the triangle, the orange triangle here is Castile, the green triangle there is Campus, And then these, the circles are all the DH lines from this population. And then the colour of is just whether they have this C2 FLC or not. So orange, they lack the C2 FLC, and the green, they have it. And you can see this clear correlation that the early flowering lines are highly enriched in these varieties which have the A2 FLCs, and the late flowering lines are enriched in lines that have at least one copy of the C2 FLC, right? And that's important. So the question is then, is it actually just this simple, like to breed for resilience? Like, I know breeding is very complicated, and I'm no breeder, but I can do a cross, right, and make double haploids. That's not a problem. So here's a field trial of this population, and the drone flyover is at a certain time of year, which enables you to distinguish Campus and Castile based on their phenology. So Castile is slightly early flowering because it's got a slightly lower chilling requirement. And you can see it flowering nicely there, and there's Campus there on the left, and you can go around. And we became interested in one particular line here, this DH28, which is a boring name. We could give it a better name, but right now it's DH28. And you can see, I hope, if you look round the trial, that the phenology of DH28 robustly matches that of Castile and not Campus. But then, if we look at the plot in the bottom right-hand corner, at the yields of these different lines, you can see the difference between Campus and Castile. They're either side of this one detectable yield gain event that we can find in the last 40 years of rapeseed breeding. And you can see DH28, this yield is not importantly different from Campus. So it has the phenology of Castile, but the yield of Campus, which is the type of resilient, it's the type of variety we're looking for. Now, of course, if you're going to do this breeding for real, not just for scientific purposes, you're going to be interested in loading these varieties with a whole bunch of other traits which I've completely ignored, so resistances. So this is not a finished variety by any means, but it quite importantly combines some of these traits from Castile with Campus without a yield penalty. And you can see this is what the plots look like at that time of year. And we can also get back out our warming trials and go through and ask, you know, does this DH28 and Castile, for that matter, do they behave any differently to standard varieties? And so one thing we can do in these warming trials is that we can measure hormone levels in the buds, and you can see the warmed. Remember, we're warming by about 5 degrees Celsius. It's not really that much, but it makes a massive difference to the ABA level in the bud of Campus. A massive difference. So you see, Campus has this mild warming, a big difference in ABA. And this has nothing to do with any drought. You know, the soil's saturated, all that. I can't emphasize that enough. And in Castile, you can see the difference in ABA is much smaller. And in DH28, you can see it has a Castile-like difference in ABA levels caused by the warming. And this is what we believe is at the heart of this resilience trait, is the fact that when the temperature varies, it is not causing variation in ABA level. And higher ABA will cause all sorts of things like failure of pollen dehiscence, all sorts of pollen problems. It'll increased grain abortion after fertilization. For instance, that's all controlled by ABA. That's particularly important in wheat because in wheat, yield is mainly determined by grain abortion. And you can see this developmentally in the apex. You see the pictures on the right. You see these warmed ones, these warm campus apex, they're developmentally retarded, and the reason is because ABA inhibits growth, right? And that's how it causes dormancy. Right. You can see I'm getting near the end, but importantly, what we also find is that there are yield differences in campus, which we don't find in Castile and we don't find in DH28. So, I have a yellow card, and this means it's time to wrap up. So, if you're interested in any of this work, then much of it is published, some of it is published. What is not published is the Castile campus story, which we're wrapping up some loose ends. We're now sort of editing the C2 in campus just to check that it really is the gene that's important, for instance. And you can look up some of these papers. They're all free to access, and we've been able to identify, although I've just talked about one, up to about 5 loci, which are in this table here, which are useful for breeding chilling resistance in winter rapeseed. So if you include any of these alleles, you either lower the chilling requirement or you make the chilling more efficient. Really interesting one is this PHP gene, which basically makes all the FLCs more sensitive to chilling. And there are other things at foot here. So one of the things that chilling does is affects the size of seeds, because the size of seeds is controlled by ABA. I haven't had time to go into that today. So also alleles that affect the size of seed are very important for resilience, we think. So what I'm going to try and suggest is that this is a new sort of type, really, of oilseed rape. It sort of combines elements of European winter phenology with elements of this sort of Chinese semi-winter, which have a very low chilling requirement. Now, all of these have a short critical photoperiod for flowering, but it has a sort of low chilling requirement, but not as low as these Chinese lines. And we found ways to differentiate between all these. So we call it European semi-winter. We think these European semi-winter type varieties will be exactly what we need to breed for climate change resilience in the UK. So, to conclude, the adaptation of oilseed rape we think is likely possible. My main message is carry on breeding your rapeseed, it's going to be fine. But don't try and model these varieties with crop models, because it won't work. And the reason it won't work is because crop models don't know about bud dormancy, because no one's put it in. Because we didn't know it existed, and likely this is not an isolated example. There'll be other things, and so it's really important to treat crop models as hypotheses for testing rather than just accepting the output. So, to conclude, the people in green the people that did the work, but I particularly want to highlight Carmel O'Neill that developed the field warming system. And Tom Locke, who's a really talented PhD student in the lab, very good brassica genomicist, if anyone is looking for one, who did all the campus Castile breeding that I talked about in the second half of the talk. So thank you for your attention. Hope you enjoyed it.